In earlier segment, we learned about prokaryotic cell and its organelles. Now we'll understand about eukaryotic cell and its organelles. A eukaryotic organism is made up with eukaryotic cells which has complex structures enclosed within the membrane. The presence of a nucleus gives eukaryotes their name, which comes from the Greek where eu means true and karyon states nucleus. Most eukaryotic cells include plant cells and animal cells, which also contain other membrane-bound organelles such as mitochondria, endoplasmic reticulum, chloroplast and Golgi apparatus. All species of large complex organisms are eukaryotes, including animals, plants and fungi, although most species of eukaryotic protists are microorganisms. Eukaryotic cells are typically much larger than those of prokaryotes. They have a variety of cytomembrane networks, membrane-bound compartments and structures called as organelle. It has a cytoskeleton composed of microtubules, microfilaments and intermediate filaments. Eukaryotic organelles are independent, membrane-bound structures or compartments which perform specific functions. These functions of organelles vary widely and typically depend on their proximity within the cell as well as their physical characteristics. Eukaryotic DNA is comprised of chromatin and is a nucleoprotein complex. A highly condensed form of chromatin is called as chromosomes, which are separated by the microtubular spindle during the cell division. Now let's make the eukaryotic cell study more interesting. Think of a cell as a microscopic city. Like a real city, it requires many services to keep it clean and run smoothly. Think of some services a real city needs, say for example traffic control, waste disposal and authoritative figure, just to name few. Like our imagined city, a cell needs the same services. Organelles are the departments that provide these services and cytomembrane networks link all other departments like routes in the city. Let's study the various functions of some common organelles. Cell wall. Now we'll have a look at cell wall. A city has a wall, similarly a cell has a cell wall which refers to its boundary, while plant cells have both cell wall and cell membrane. Animal cells are devoid of cell wall. The cell wall is tough, usually flexible but sometimes fairly rigid layer. It is located outside the cell membrane and provides structural support, protection and also acts as a filtering mechanism. A major function of cell wall is preventing overexpansion when water enters the cell. It is formed of multiple layers of carbohydrates, mainly cellulose and pectin, which gives structural support, rigidity, strength and protection against mechanical stress to the cells. It permits the organism to build and hold its shape. The cell wall also limits the entry of large molecules that may be toxic to the cell. Plasma membrane. A plasma membrane can be named as a cell boundary in common language, just like a city has a city border. Cell membrane can be termed as a plasma membrane. S.J. Singer and Garth Nicholson in 1972 described a model for plasma membrane known as fluid mosaic model. In electron micrographs, prokaryotic and eukaryotic plasma membrane look like two layered structures. The phospholipid molecules are arranged in two parallel rows, which is also called as a lipid bilayer. Each phospholipid molecules contains a polar head composed of hydrophilic phosphate group and glycerol, and hydrophobic nonpolar tails are composed of fatty acids. The polar heads are on the surface of the lipid bilayer and the non-polar tails are in the interior of the bilayer. Proteins are also present in the structure of plasma membrane. On the basis of its position, they are divided into two types. First, the extrinsic membrane proteins, second, the intrinsic membrane proteins. Now, let's have a look at the functions of plasma membrane. It is the membrane boundary of the cell which regulates cell transport. The plasma membrane exhibits selective permeability. Small non-polar hydrophobic molecules are soluble in lipids and can easily pass through the membrane. Polar hydrophilic molecules are not soluble in lipids. Transport of these polar molecules across the membrane depends on the protein molecules in the phospholipid bilayer. Cytoplasm. The cytoplasm acts as lawns and parks of the cell. Cytoplasm is a semi-fluid substance found inside the cell. The cytoplasm protects the internal organelles and provides medium for diffusion. It is the cell landscape found in any space where organelles are not present and therefore is much like the lawns and parks of our city. It is within the cytoplasm that most cellular activities occur, such as almost all metabolic pathways, including glycolysis and other processes such as endocytosis and exocytosis, as well as cell division. Cytoskeleton. Let's take a look at cytoskeleton. Cytoskeleton can be called as steel girders of the cell. The cytoskeleton makes up the internal framework, like the steel girders that are the framework for the buildings in the city, that gives each cell with its distinctive shape and high level of organization. The cytoskeleton consists of a web or mesh of protein fibers that pass throughout the cell, forming the cell shape, gives cell support, manages intracellular traffic, and important for locomotion. The cell materials flow within the paths defined by the membrane walls and tubules, and traffic from one organelle to another is highly regulated. The three types of protein filaments found in cytoskeleton include the microfilaments, microtubules, and the intermediate filaments. These perform a variety of specific cell processes and functions. Microtubules are hollow tubes of tubulin which enhances structural support to organelles for movement. Microfilaments are solid rod-like structures of actin, acts as structural support. The interlinker filaments link all components which are called as microtubular lattice. Ribosomes. Ribosomes are better known as a lumber or brickyard of the cell. Ribosomes are granular organelles composed of RNA and protein which helps in synthesizing proteins, consists of small and large unit. These units are attached to mRNA. The ribosomes carry out manual labor in the form of protein synthesis for the nucleus. They bring together all the raw ingredients such as RNA and amino acids to assemble proteins. The proteins created are essential to the cell and its function. Think of proteins as machinery for cell functions, much like the electricity and plumbing are essential for the real city. For example, enzymes are type of proteins without which life could not exist. Endoplasmic reticulum. Now let's take a look at endoplasmic reticulum and its types. Endoplasmic reticulum is the highways and road system of the cell. Goods or molecules which are manufactured by the organelles and cytoplasm can be shipped to the needed areas via this road system. The endoplasmic reticulum was discovered in 1945 by Albert Claude and Keith Potter. It was first noticed in the cytoplasm of chick embryo cells. The endoplasmic reticulum extends away from nuclear envelope through a network of tubules. There are two types of endoplasmic reticulum. First, smooth endoplasmic reticulum, and second is rough endoplasmic reticulum. This extensive network makes up approximately 1/2 of all membranous tissue of the cell and is the site of membrane and protein synthesis. Rough endoplasmic reticulum is named for the presence of ribosomes on its surface, and it is the site of protein synthesis, while the smooth endoplasmic reticulum lacks ribosomes and is responsible for lipid synthesis and synthesis of other metabolic processes such as drug detoxification. Golgi bodies. It has been named after Italian microscopist Camillo Golgi, who announced this discovery in 1898. The membranous sac which reside near the nucleus and endoplasmic reticulum are known as Golgi apparatus. The Golgi apparatus performs several functions in close partnership with the endoplasmic reticulum. The Golgi apparatus receives and modifies the substances that are manufactured by the endoplasmic reticulum. One side of Golgi serves as a receiving dock for vesicles produced by the ER. Once in the Golgi, the materials in the vesicles are modified with the assembly of carbohydrates with proteins or lipids with proteins. The opposite side of the Golgi serves as a shipping depot. Finished products are sorted out and are bound in the vesicles, which move towards the cell membrane for export. They may also move into other organelles. As the contents pass through the organelle, any appropriate chemical alterations are performed. The contents may be then transported inside or outside the cell as deemed necessary. Mitochondria. In 1886, a cytologist named Richard Altman termed mitochondria as bioblast. Then in 1898, Carl Benda renamed the term bioblast as mitochondria, which was taken from the Greek words mitos means thread and chondros states granules. Mitochondria are considered as energy plant of the cell. Mitochondria in cell are like power plant in the city. Mitochondria contain their own DNA. The mitochondrion is bound by two membranes, outer membrane and inner membrane, each made up with phospholipid bilayer. Outer membrane covers intermembrane space and inner membrane covers matrix. This matrix is also known as mitochondrial matrix. The inner membrane is highly folded and has enzymes embedded in it that are responsible for the production of ATP through electron transport chain known as aerobic respiration. Each fold is called cristae as they greatly increase the membrane's surface area for ATP production and shows varied structural forms. Mitochondrion metabolizes carbohydrates, fatty acids and amino acids into oxidative energy required to sustain the eukaryotic cell. The mitochondrion makes possible the energy transduction of the cells. Mitochondria carry out the processes of cellular respiration in which the chemical energy of food such as sugars and other biomolecules are metabolized and is converted into chemical energy of the cellular fuel molecule called as ATP. Chloroplast. The chloroplast can also be known as a solar energy plant. Konstantin Merezhkovsky, a noted Russian botanist and biologist, discovered chloroplast in 1905. Chloroplasts are found primarily in algae and plants and some protists. The three major internal membrane partitions of chloroplast are the intermembrane space between the outer membrane and the inner membranes. They have highly permeable outer membrane, a much less permeable inner membrane in which membrane transport proteins are embedded, and a narrow intermembrane space between them. The space enclosed by the inner membrane, which contains a fluid and a network of tubules and hollow discs formed of a membrane, is called as stroma. The space inside the membranous tubules and discs are called as thylakoids. The discs that occur as stacks are called as granum. At this site, the chlorophyll actually traps the solar energy. Like mitochondria, the chloroplast also contain its own DNA, which is responsible for the production of different regulatory proteins. The DNA which is present in the organelles like mitochondria or chloroplast as well as in the cytoplasm of some protists are known as extrachromosomal or extranuclear or cytoplasmic genetic material and are hence responsible for maternal inheritance. Chloroplasts are complex structures that carry out photosynthesis, absorbing solar energy from the sun and convert it into chemical energy in the sugar molecules through a process known as photosynthesis, evolving a byproduct oxygen. It provides chemical energy directly or indirectly for all other living organisms. Hence, chloroplasts are unique organelles because they are capable of capturing, converting and conserving solar energy in the form of chemical energy. Nucleus. The nucleus acts as a mayor of the cell which gives the orders to perform various functions like the mayor of the city. The nucleus was for years the criteria required for an organism to be eukaryote. Thonius Philip van Leeuwenhoek, a Dutch scientist, first observed the nucleus within the red blood cells of fish, but the term cell nucleus was given by Robert Brown, a Scottish botanist. Much like the brain of humans, the nucleus represents the control center for the cell, which consists of cellular proteins, DNA and RNA. Using DNA as a blueprint, like the blueprints of the city, the nucleus directs the production of proteins. Prokaryotes usually contain one circular molecule of DNA, which represents its genetic material or genome, while eukaryotes' genome is structured into the number of linear chromosomes. The nuclear envelope is double membrane, consists of a nuclear pore complex which represents the outer limits of the nucleus. Nucleolus is a granular body within the nucleus which is the site of rRNA synthesis. It also consists of chromosomes which are composed of DNA protein complex and contains genes. Centrioles. A centriole is a barrel-shaped cell structure found in most animal eukaryotic cells and is absent in higher plants and most fungi. They help to organize the assembly of microtubules during cell division. Centrioles replicate during the interphase stage of mitosis and meiosis. Centrioles are called as basal bodies from cilia and flagella. The centrosome, also called as the microtubule organizing center, is an area in the cell where microtubules are produced. Within an animal cell's centrosome, there is a pair of small organelles, the centrioles, each made up of a ring of 9 groups of microtubules. There are 3 fused microtubules in each group. The 2 centrioles are arranged such that the one is perpendicular to other. During animal cell division, the centrosome divides and the centrioles replicate. The result is 2 centrosomes, each with its own pair of centrioles. The 2 centrosomes move to opposite ends of the nucleus, and from each centrosome, microtubules grow into a spindle which is responsible for separating replicated chromosomes into the 2 daughter cells. While plant cells have centrosomes that function much like animal cell centrosomes, but these do not have centrioles. Lysosomes. Lysosomes are waste disposal and a recycling unit of the cell. They are produced by the rough endoplasmic reticulum and the Golgi apparatus. The lysosomes are digestive sacs that can break down macromolecules in the cell using the process of hydrolysis. The digestion is carried out with lysosomal enzymes found in the lysosome. Like waste disposal in a city, lysosome helps in the disposal of waste material in the cell. The lysosomes also serve as recycling centers for the cell by engulfing old organelles by autophage and making its molecules available for the formation of new organelles. Vacuoles and microbodies. The vacuoles and vesicles can play the role of warehouses, water towers or garbage dumps of the cell. Vacuoles are membranous sacs which transport and store water and other materials. They can hold many substances from organic molecules to simple excess water. There are three types of vacuoles, namely food vacuole, central vacuole and contractile vacuole. Food vacuoles store food taken in by the cells and later works in conjunction with the lysosomes. Central vacuole in plants serve as a large lysosome which help plants grow in size and store vital chemicals as well as maintaining plant turbidity. Contractile vacuoles are found in number of protists and play a role in osmoregulation. While the microbodies are cytoplasmic organelles of a more or less globular shape that comprises of degradative enzymes for metabolic activities bound within a single membrane. Peroxisomes. Peroxisomes are found in all eukaryotic cells. They contain oxidative enzymes such as catalase and urate oxidase. Like mitochondria, peroxisomes are major sites of oxygen utilization. One hypothesis is that peroxisomes are vestige of an ancient organelle that had performed entire oxygen metabolism in the primitive ancestors of eukaryotic cells. Peroxisomes differ from mitochondria and chloroplast in many ways. Notably, they are surrounded by only a single membrane and they do not contain DNA or ribosomes. Like mitochondria and chloroplast, however, peroxisomes are thought to acquire their proteins by selective import from the cytosol. Because they have no genome, all of their proteins must be imported. Peroxisomes thus resemble the endoplasmic reticulum in being a self-replicating membrane-enclosed organelle that exists without a genome of its own. Now, we have understood the complexity of eukaryotic cell as compared to prokaryotic cell. The main feature of eukaryotic cell is the presence of well-developed nuclear envelope bound nucleus. The membrane-bound organelles are also present as cytoplasmic inclusion bodies, which is a distinguished feature. Now we have studied that eukaryotic cell is divided into two major categories, namely eukaryotic plant cell and eukaryotic animal cell. The difference between them is the plant cell consists of cell wall and chloroplast, while generally animal cell are devoid of these features.